Okay, so our final speaker will be Jaqueline Kirchner from the Gates Foundation, and the title of her talk will be Developing Anti-Infection MAbs for Malaria, Implications for Global Health.
I'd like to thank Bob and Josh very much for the invitation to speak to you today. It's a fantastic opportunity to tell you a little bit about our strategy for malaria eradication at the Bill and Melinda Gates Foundation, and what we think will be a critical role for a monoclonal antibody to prevent infection within that strategy. Before I begin, I just want to make the point that nothing we do at the foundation is by ourselves. In fact, we have no labs, we don't own anything, we don't do any actual work ourselves. We do this all through fantastic partners who are really smart, motivated people who are equally eager to see the end of malaria. So just want to make that point that we really are grateful for our interactions with the field, with the community, and would welcome more. In addition, I'm here representing a team of people at the foundation. We have people that are, I'm getting a time, oh, you have to start the clock. I get an extra minute. I shouldn't have told you that. I'm representing a team here. I'm in the Discovery and Translational Sciences team. We also have people in the Malaria Program Strategy team here at the meeting that you can speak with. We also have colleagues at a newly established Gates Medical Research Institute, which is a product development partner for the foundation that is working on not only malaria, but also TB and enteric diarrheal diseases. So we began a lot of our talks at the foundation with a problem statement, and this is to really help us focus on what are the key issues in global health that we need to address, where we can have the greatest impact. And this is information that I'm sure everybody in this room is very familiar with, but just to kind of set the stage, remind you, why malaria, why now, and why monoclonal antibodies? So as you can see from this report that just came out in the 2018 World Malaria Report, there's been a substantial decrease in malaria cases over the last many years, largely due to vector control. But that curve seems to be flattening and may, even more worrying, be starting to uptick. So the question is, are we actually seeing a rebound? That's one point. And then in addition to just looking at the malaria cases, although the number of deaths due to malaria is still decreasing, it appears that that curve is flattening as well. So this really cries out for a need for more tools. And our strategy at the foundation is to eradicate malaria by 2040 through a heavy emphasis on interventions that reduce burden while preventing transmission. So how are we planning to do that? We have a very diverse set of investments in different R&D tools. Those are listed here. In addition to diagnostics and vector control solutions, drugs for case management, none of which I will be talking about today. We have a series of investments in prevention of infection. This is mostly focused on prevention of Plasmodium falciparum. And the foundation is well known for work on vaccines, but in addition to vaccines, we're also investing in long-acting small molecules for prevention and in monoclonal antibodies. And I think people have raised a number of very reasonable and realistic concerns about a monoclonal antibody approach for infectious disease generally. And then to add on top of that, how are we planning to do this for global health, where cost is a huge factor? So I can tell you that I feel like a little schizophrenic, like these two screens here. I feel both skeptical as a scientist, as well as very optimistic that this is an important part of the strategy to add to our arsenal of tools. There's risk with every one of these strategies. None of them is a slam dunk. And what we've learned from trying to eradicate polio is that we do want to have more tools in our arsenal so that when we get to that last mile, which we're experiencing right now with polio, we don't run out of tools. We have more things we can turn to and more approaches we can try to get to eradication. In terms of the cost of a monoclonal antibody and the utility in infectious disease, there's little precedent for a clinically approved monoclonal for infectious disease. In fact, I can only think of RSV antibody, which is quite effective. However, there's more to be done, and there's a lot of improvements in the technology. And one of the investments we've also made is trying to reduce the cost of manufacturing through investment in a biotech company called Just Biotherapeutics, which is absolutely focused on bringing the cost of monoclonals down to industry disrupting standards. So we are well aware of the risks. We are well aware of the strategic issues with this approach, but we think that long-acting small molecules run the risk of resistance to those drugs. Vaccines, we've already heard from Bob very nicely, laid out some of the challenges with getting a good vaccine in in this disease through hyperresponsive issues, as well as the challenge of getting durability protection. So this is yet another tool. It's a risk mitigation approach, and we think that all of these approaches are worthwhile investing in. So village-based vector control and case management, while it's been effective so far, it's unlikely sufficient to continue to provide the same returns on investment, and especially for getting to eradication in places where malaria transmission is high, coverage and compliance with household-based vector controls is poor. There's social behavior such as being outside, leaving the village and coming back to the village. So one of the issues with trying to get to eradication, even if you had a perfect tool, is that in areas where malaria has been eliminated, there's still an issue with human migration, not only in and out of villages, but also across transnational borders that we think about. And so this brings us to a way to think about how to use these strategies in an effective manner. So we've identified 5 distinct use cases for seasonal prevention of malaria. I'm just going to load this all. The first is infants and children. And with infants and children, clearly this is the population that has been identified by the WHO for SMC, for chemoprevention. This is where the vast majority of the burden of malaria exists. We are proposing to provide prevention measures in infants and children to cover from 3 months to 10 years, which is beyond what the WHO has currently approved for SMC. We know that SMC is effective. There's no question that it can be very effective, but the reality is that while it's up to 80% effective in the clinic, it can be as low as 30% effective in the field due to some issues with coverage and compliance. Only about 50% of children that are eligible for SMC have access to it, so there are some significant hurdles with that approach currently. Pregnant women, we want to reduce malaria burden in women by preventing infection and placental malaria in prima gravids, and reduce the fetal risks associated with placental malaria in all pregnancies. We want to go after high-risk workers. These are people who may be resistant to severe malaria, but are still capable of transmitting malaria. And as they leave the village and come back to the village, they may leave a village that's been cleared of malaria, but bring back in infectious parasitemia. So this is another population that we'd like to provide preventive measures to. And then there's crisis situations, so preventing and prophylaxing against Plasmodium falciparum malaria in crisis scenarios. This is something that has been seen when there's been outbreaks of Ebola. Suddenly, SMC delivery has become a huge challenge, so having a way to provide protection through those type of crisis situations is quite important. So these first 4 use cases are in geographies and populations that are relevant to our foundation strategy and are high priority. The last one is out of scope for our foundation strategy, but I did want to mention this because it's already come up. Travelers and military, other short-term workers that are deployed into the field that are immunologically naive, not malaria exposed, but need to be protected. This provides a different use case where the prevention of infection would have to be very high, would have to be as good as Malarone, for example. And it's not something that is within our strategy, but we also see it as a potential dual market opportunity that might be attractive to some commercial entities. So I just wanted to mention that. So these use cases can be solved, intervened in by 3 different approaches as shown here. Long-acting chemoprevention in the form of injectable small molecules, monoclonal antibodies, and vaccines. We do have substantial investments and efforts in all 3 of these areas. And as you can see, if you look at what the delivery might be over different rainy seasons and what the protection might be, they offer different advantages and disadvantages. The differentiation of these will come down to how long the prophylactic tail is, the ease of use and delivery, and in fact, the cost effectiveness. In all 3 cases, what will likely need to be the case will be delivery of a curative dose of an appropriate antimalarial to clear any ongoing existing parasitemia, followed by one of these preventive measures to protect from reinfection. So what is our monoclonal antibody strategy? You've already seen the slide, and Bob very nicely went through it and pointed out some of the important points that I want to reiterate here, just to bookend this concept. When you look at the malaria life cycle, there's a number of attractive places that you could intervene. A number of places you could imagine a monoclonal antibody might work very well. It is far more difficult than any monoclonal antibody that's been clinically approved, which generally disrupts the protein-protein interaction. We don't think right now that the antibodies that are effective in malaria, there's no evidence that all they're doing is a simple protein-protein disruption. So we're actually asking a lot mechanistically out of these antibodies that has not been asked of the over 200 clinically approved monoclonal antibodies for autoimmune inflammatory disease or immuno-oncology. So it is a challenging situation, and because of that, when you think about it from a drug development perspective, you sort of look for the place where there's the least amount of target that you need to cover with your antibody. And the obvious place here is in the pre-erythrocytic stage at the point of the sporozoite. It's where the least amount of material is present in the host that the antibody must bind to and have its activity against. After that stage, as Bob has already told you and as Simon alluded to as well, there is an amplification of the target, and while it might be possible to inhibit those stages, it certainly isn't the easiest place to start. Our strategy is really focused on the pre-erythrocytic stage. After that point, our concern is that if we were blocking at a blood stage, while it might be quite possible to do that, you may reduce the disease in individuals, but they may be asymptomatic and parasitemic, and therefore would not actually contribute to our overall prevention of transmission strategy. So within that sporozoite target class, Josh has nicely laid out that there may be additional sporozoite targets that could be of interest, and we certainly eagerly anticipate the identification of those targets. But right now, again, as hard as it is, the low-hanging fruit is the CSP protein. This is a very nice situation in a drug development paradigm where you have a clinically validated target. So we know that it's the dominant surface protein. The epitope is conserved amongst all the Plasmodium falciparum strains. It's clinically validated by the RTSS vaccine efficacy data. There's preclinical validation for a monoclonal antibody approach in a number of different mouse challenge models from many different groups using many different antibodies. So that's also a very nice situation to be in. And we are eagerly anticipating clinical proof of concept data when CIS43 goes into the clinic next year. So that's all very good. And while we're very hopeful that we'll see efficacy with CIS43, we also want to be very pragmatic and continue to look for additional monoclonal antibodies which may be incrementally more potent, but hopefully far more potent than that, so we can use that highly potent antibody to bring down the cost of goods. So how are we planning to do this? We are casting a wide net and being quite open to monoclonal antibodies coming from whatever source they come from. We're working with both academic investigators as well as for-profits, and we're looking for the antibodies to demonstrate their potency in standardized assays with benchmark controls in single locations so that we can understand the relative potency and the relative characteristics of these antibodies coming out of standardized assays. That allows us to make data-driven decisions about which antibodies might be the most potent ones and most likely to have success going into the clinic. So these are going into affinity measurements against full-length CSP and individual peptides that are known to be important epitopes within the repeat region of the CSP protein. They also go into the sporozoite hepatocyte traversal assay. And we've already been told, and there's data in the literature showing that there's not a complete one-to-one correlation between that in vitro assay and the in vivo function of an antibody. But we believe that it's important to get a complete data set on a large number of antibodies so we can start to understand what are the rules that make a good potent antibody. And when we understand those rules better, then we can better iterate on how to optimize an antibody for clinical efficacy. The antibodies which are most potent in the liver burden model and the bite parasitemia model, which are being run in Fidel Zavala's lab, will then go on to undergo molecular optimization of the sequences, including variants that make them more developable as a drug, go into cell line development, and in parallel, we will confirm the activity in the FRG humanized mouse model, as this is the only challenge model that uses a wild-type version of Plasmodium falciparum preclinically. And the antibodies will all be considered very carefully. Some number of lead candidates will then go into IND-enabling studies and be taken into the clinic for proof of concept in controlled human malaria infection model. And right now we are planning to do those studies with our product development partners at the Gates Medical Research Institute. Okay, so how potent does the antibody need to be? This is a really critical part of the target product profile, and I want to spend just a little bit of time on this. This slide is sort of dense, but I'll talk you through it. Even before you know what the target is or what assay the antibody has been in, without knowing any of that, if you just go by what information we already have about monoclonal antibodies in humans and their pharmacokinetic profile, and given that we want to achieve protection for at least 3 months, hopefully for 6 months, we have certain constraints on the system. There's a solubility issue with monoclonal antibodies. Generally, you can get them to be about 100 to 125 mg per mil. Sometimes a little higher, but you don't want to count on that. Given that concentration of antibody and the volume constraints with an intramuscular or subcutaneous delivery, which is what we would be using in the geographies we're planning to take this to, with all those constraints taken together, looking at the pharmacokinetic profiles in an infant, which is about 7 kilograms, a child, about 20 kilograms, or an adult, which is 100 kilograms, you can predict for an antibody with a half-life extension mutation that Bob alluded to earlier, what the concentrations in serum will be at the end of 3 months and at the end of 6 months for a well-behaved antibody. These are knowns. These are things you can predict. Given all of those constraints, that anticipated pharmacokinetic profile, that actually translates to, for protection for 3 months, the antibody must be effective at about 13 micrograms per mil, and for protection at 6 months, even lower than that. So this is a high bar. We're asking a lot out of the antibody, and it helps to also think about how we use our preclinical assays for predicting clinical efficacy. Now, we don't yet know how any of those mouse models translates to the clinic. With the extremely valuable data that Bob and the VRC will be generating with the CIS43 antibody, we'll start to get some insight into that, and that will help us to go back and understand whether antibodies in those preclinical models might actually be successful in the clinic or not. But I just want to make this point because it is really the case that the antibodies need to be effective at low concentrations in the serum to meet the TPP. So we spent quite a lot of time with a lot of experts in the field to form an interventional target product profile for a monoclonal antibody for malaria. And this is available. I can send it to anybody. I think the slides will be available after this. It's too small to read. So I just want to put that up there. This is our standard TPP for a monoclonal antibody for malaria. But to give you an easier way to sort of get the information, condensed it here. So the plan is for seasonal prevention of Plasmodium falciparum for 3 to 6 months. We require no more than 2 antibodies due to cost, but ideally it would be 1 antibody, and this could even be a bispecific. The plan is to go into children for as young as 3 months and as old as 10 years and everywhere in between. That's the base case. We would ideally like the antibody to be available to protect adults as well in the use cases that I described earlier. We will use this in moderate to high transmission settings with seasonal pattern of transmission and outbreaks. And the clinical efficacy requirements are 80% to achieve non-inferiority to SMC. And of course, it has to be as safe as SMC, and hopefully maybe even safer than SMC. For the travelers and military market, for non-immune individuals, non-malaria exposed, we would require a much higher efficacy, 98%, to achieve non-inferiority to Malarone. The potency requirements I just described to you are 3 to 13 micrograms per mil in serum. This would require not only a potent antibody, but an antibody with a half-life extension mutation. We don't really have any confidence that we can routinely give in a cost-effective manner IV infusion in the geographies that we're interested in bringing an antibody to, so we require that this would be administered by IM or sub-Q. 1.5 to 2 mils for infants and children, and 1 to 1.5 mils to adults before the start of the rainy season. So how do we make antibodies that meet that TPP? First of all, how do we make antibodies that are affordable? So again, we've made investments in CMOs to lower the cost of manufacturing, and this can be done with higher yields, process improvements. It can also be done by improving the PK. In addition to the known half-life extension mutations, there may be other half-life extension mutations and formulation considerations that can increase the concentration of the antibody and improve the potency. But honestly, one of the best levers to reduce your cost of goods is to have a much more potent antibody. So the more potent it is, the less you need to give, and that reduces your cost of goods tremendously. But how do we do that? So one hypothesis is that we could increase the affinity and the avidity for the CSP protein. There's some evidence that that will help. Will that be enough? Maybe, maybe not. What about enhancing effector function? We already know that effector function in the form of binding to Fc receptor is not required for all CSP monoclonal antibodies, but it might be a useful attribute to have in some, and if we can harness the host immune system to provide additional mechanisms of destruction of the sporozoite, that might be a huge advantage. It might shift the potency curve to the left. We're considering bispecific and multimeric structures. We're considering antibody-drug conjugates, and happy to hear other ideas. What about other pre-erythrocyte targets? I think it's fantastic that Josh and others are going after non-CSP sporozoite targets. These would be of great interest. The epitope does need, for our purposes, the epitope needs to be conserved among different strains of falciparum. Cross-reactivity to other species like Vivax would be a huge advantage, but not required. The merozoite target, very attractive, very interesting. The proof of concept data looks extremely intriguing. I think what we really need to see is that for the TPP, a demonstration that it could provide sterilizing protection on its own or rescue a partially protective anti-CSP antibody. Okay, and lastly, I'd just like to acknowledge we do all of this work, as I said, with partners. We've had fantastic partnerships over the last couple of years as we've been working on monoclonal antibodies for malaria and hope to have many more. And be happy to take any questions.
Michael.
Thank you very much. I was interested in your target product profile. You're looking for, as I understand it, for the at the pre-erythrocytic stage for something that's effective for 3 to 6 months with about 80% efficacy or thereabouts. How does that compare to what's required for RTSS?
How does that compare to what's required?
Yeah, I mean, RTSS gives you about 80% efficacy over the first 3 to 6 months, after which time it drops off dramatically. But for those first 3 to 6 months, that's the level of protection you're seeing.
Yeah, so again, I think for RTSS, what we are trying to do with the next-gen CSP vaccine effort is to come up with a vaccine that can overcome some of the hyper-responsiveness that's seen in malaria-exposed individuals and provide a longer durability of protection. So it's not an either-or scenario. I think that we still want to find a better vaccine. And then the monoclonal antibody would be immediate protection, so you would not require the individual to mount an immune response to the immunogen, but it provides protection and overcomes the hurdle of having to mount an immune response. So for those individuals who don't mount a good response to a vaccine, this would provide another way to provide that sort of longer-term protection.
Sure, but in that first 3 to 6 months, which is a critical period for the monoclonal, it will come down to a cost cent. How much does RTSS cost versus how much the monoclonal costs?
Yeah, that's a good question. So I have to tell you that we've not yet determined the appropriate cost of goods for this in the target product profile. There's been a lot of discussion around that. These kinds of issues do come up in those conversations, and I think we're still working through that.
Thanks. Michael, RTSS would require 3 immunizations. So you have 3 immunizations, 0, 4, and 8. It's only done in 5 to 17-month-olds, so how would it work in older people? And then you have the clinic time of giving multiple immunizations. So I think this is an efficiency because you're proposing only to give a single passive transfer.
Bob, you're right, but it does assume that antibody will last that long. That's what it comes down to, and you haven't shown
She
that
laid
yet.
it out. It's potency, right, and pharmacokinetics, if they last that long.
If you have to give that monoclonal more than once, you're no different to RTSS for that first 3 to 6 months.
That's right. So that's why the TPP is minimum is 3 months, but ideally we're shooting for 6 months.
Thank you very much. I just have a comment. I mean, in addition to the skepticism that I already voiced, I mean, I would be particularly skeptic actually to try to do intervention against placental malaria by seasonal malaria control by monoclonal antibodies. I think there is now a rapidly increasing body of evidence to suggest that the problem of malaria in pregnancy, placental malaria, is really a problem of women who already carry an asymptomatic infection becoming pregnant. rather than the problem of pregnant women becoming infected during pregnancy. So there is actually very little seasonality of placental malaria, in contrast to other forms of malaria in areas where the malaria transmission is seasonal. So I think I would find it very unlikely that short-term prophylaxis by monoclonal antibodies against placental malaria, even if you had a very effective monoclonal antibody, would actually make any difference.
Thank you for that comment. So I do think that it's important to just reiterate that the strategy here is to not give these preventions without first clearing parasitemia. So the plan is to clear parasitemia and then provide the prevention, whether it's the long-acting small molecules, the antibody, or the vaccine. In this case, we think that there's a real interest in using the antibody in pregnant mothers because it may offer safety advantages over IPTP.
But that, I think it could possibly work as a therapeutic in pregnant women, possibly, but I'm just cautioning against using it as a seasonal malaria prevention, where you will use it as a blanket. The problem really with malaria and pregnancy is that the women only come to the clinic, come into contact with the health system at a point where the damage is already done. Right?
Yeah, we've had discussions about how to actually make this work, and we talked about, is there a way we could bring the antibody to adolescent girls, women of reproductive age, and that's still an ongoing discussion. We have other efforts of the foundation with HPV that we may be able to follow in the footsteps of that, but I think it's a really good point.
Yeah. Okay. Well, thank you very much, and thank all the speakers.